All right, everyone, we're going to get started here today. So welcome everyone to the first lightning talk of this year. I'm Dustin. I'm a third-year PhD student in Biostats. Hey, now. So for those of you who haven't been to lightning talks before, basically the setup is we're going to have 4 speakers. They're going to speak for less than or equal to 5 minutes each, and they're going to be cut off by a very creative buzzer. And then after their talk, they're going to have about 1 minute for any questions, like 1 or 2 quick questions, and then obviously we have time afterwards to ask more in-depth questions. And then hang around afterwards to grab food, more drinks, and to mingle. So let's welcome our first speaker, Michele, who's a 4th-year PhD student and the president of the BSCC, which she'll tell you more about now.
All right, so thank you, Dustin. Before jumping into the Biostat Student Consulting Center, I just want to tell you a little bit about myself. So like you said, I'm a 4th-year doctoral student. I'm co-advised by Brent Kuhl and Jeff Miller. Now, you're probably understandably unfamiliar with some of my work on functional data analysis or discovery of gene regulatory networks. But those of you who may have been on the department listserv for the past 3 years or so might be more familiar with my work with my collaborator, Susan Levisi, on developing risk models for cancellation of department meetings due to forecasting errors. So, love you on that. Anyway.
Very scientific.
So, back to the topic at hand. What is the BSCC? So, we are a free, student-run, short-term statistical consulting service provided by doctoral students here in the department. We were founded in 2016 through a learning and teaching grant that was awarded to Emily Slade. And we are open to students here at HSPH as well as 14 different life sciences programs in the Longwood community. So those students can access the BSCC through 2 different ways, either through walk-in office hours held right across the hall there on Mondays and Thursdays, or by submitting an online inquiry to our website. You may be familiar with the consulting course that the department offers and that 3rd-year students often take. So while students in that course are required to be in the BSCC, they actually are 2 separate entities, and you don't have to be enrolled in the course to be a student consultant in the BSCC. So like I said, we've only been open for about 2 years, but in those 2 years, we've already received about 170 online inquiries, 85 office hours visits. And you can see the bulk of those requests are coming from students in the EPI department as well as the life sciences departments, but we receive a smattering of inquiries from students in all the other departments here at HSPH as well. So what are those cases like? Some of them have to do with questions about missing data or performing a meta-analysis, and some students come with a pretty good idea of what methods they want to use, like this first student who said, I want to use a mixed effects model to analyze some longitudinal data that I have from my master's thesis. While other students have a data set and they've done some basic manipulations, like this student here with surveys from Syrian refugees, but they don't really know how to proceed. So that's where they come to the BSCC, and a student consultant will help walk them through formulating an analysis plan. In terms of training, we have monthly meetings where we review open cases and client feedback. We also break into small groups to discuss and work through case studies. And in the last semester, we invited a few faculty members of Dr. Paul Catalano, Dr. Schoenfeld, and Dr. Kalish, shown here, to come speak to us. So if you're a faculty member or postdoc who'd like to come speak, we're always looking for people. So if you have an interesting statistical problem that you'd like to walk us through, please let us know. In terms of feedback, it's largely been very positive. When we surveyed the consultants, they said that the BSCC improved their consulting skills and that they enjoyed their experience, and it gave them a chance to actually apply the lessons that they learned in more theoretical classes. And clients in general felt that consultants solved their problems or gave them resources to do so, and that they would go back to the BSCC in the future if they had another statistical question. So even though the feedback has been largely positive, we're always looking for ways to improve consultant training. So we're seeking faculty members or postdocs who'd be willing to mentor student consultants. And we could see that working in a number of ways, but one possibility could be to invite students to attend your consulting meetings or times that you're talking to collaborators, or involving students in defining a project or formulating a statistical problem. So that level of involvement would be up to you, but if you are interested, definitely talk to someone on our leadership team. That includes Dustin, our MC, myself, Emma Thomas, and Tony Tawitai, who are also here. as well as our faculty advisor, Dr. LJ Wei. So I just want to say thank you for listening. Hopefully next time you see the BSCC pop up in the Biostat newsletter, you'll have a better understanding of who we are. And also a big thank you to the Biostats department and the administrative staff for your continued support for the BSCC. Thank you.
So any questions for Michelle? Yeah.
I'm wondering who your consultants are. Are they mostly PhDs, or are they
There
masters in our department, or are they from other departments as well?
so it's about 25 doctoral students who have passed their qualifying exams.
Okay.
Right. Yeah. It varies every semester, but we've had about 20 to 30 so far each semester.
You might think of it trying to include the master students as well under the tutelage of the doctoral students, because I think a lot of master students really would like this kind of experience.
Yeah, that's a good point. That's something we've thought about, and I think we'll continue to think about in the future.
Any other questions for Michelle? All right, if anyone has any other questions, so Michelle has to take off. She's going to hand out some
I have
pens.
a bunch of pens. We got like thousands of them. I'm going to leave them here with Dustin. If you
Yeah,
want.
so you can come talk to me, Tony, or Emma afterwards if you have other questions.
I didn't know lightning talks were supposed to have swag.
Oh, yeah. Not until now. You didn't get the memo? All right, so thank you, Michelle. Next up, we have Camila, who I've never met before, but I'm about to. All right. So Camila, as far as I know, is a postdoc at Dana-Farber.
Yes.
Not anymore.
So yeah, so was a postdoc at Dana-Farber with John Quackenbush, and now I'm starting as a research associate here at the department. So today I'm going to present the work we've developed during my postdoc, which is the work that I plan to explain here in the department. So I'm interested in using gene regulatory networks to understand sex differences. And I will put the timer so I can look at it. So first of all, why should we study sex differences? So we know there are significant sex differences during the development of many diseases, which can influence disease incidence and survival. But still today, this is a very understudied topic, and it has become a priority for the NIH. So in cancer, for example, males have a higher risk and a higher mortality rate than females. And this is true for many cancer types, but still today, males and females receive the same treatment. So if we think about it, most of the sex differences come primarily from the inequality between the sex hormones and the influence between the sex chromosomes and the influence of the sex hormones. But if we think about it, the sex difference won't be restricted to these factors. And our hypothesis is that the combination of all these sex bias factors will influence gene regulation in males and females. So what we've been doing is we have been applying gene regulatory networks to understand sex-specific gene regulation. And we have done this in colon cancer and also in 28 healthy tissues. So our method was developed by Kimberly Glass and Marika Kutcher a few years ago. So it's a network-based approach to infer the regulatory networks between transcription factors and target genes. So PANDA starts with a prior network. Well, I'll briefly say that this algorithm uses message passing approach to integrate different types of genomic data and then updates if information is flowing through transcription factor to target genes. So at the end, what we're going to have is a regulatory network that looks like this, where each edge connects a transcription factor to a target gene, and the edge weight indicates the likelihood that regulation is happening between this transcription factor and this target gene. So PANDA infers one network for a population, and then we can use Linus to generate one network for each sample. So what Linus does is first it uses PANDA to generate a network for all the samples, then it leaves out one sample and re-estimates the network. Then based on the perturbation that this missing sample, that Linus can then estimate the network for this left out sample. So at the end, we have one network for each individual, and we can compare networks. So in our case, we're comparing the female networks with the male networks. In this work, we use data from TCGA and GEO, and we generated hundreds of networks. So one way to analyze this data is we can calculate the gene targeting score. So this is the gene in degree, or this is the sum of all the edge weights a gene received from the complete set of transcription factors. Then we can compare this gene targeting score between males and females. And we do this by applying a linear model correcting for age, state, and race. So at this point, we have the genes that are being differentially targeted or differentially regulated between males and females. We can then use gene set enrichment analysis to find the pathways that are enriched for these genes. So we did this in colon cancer, and we found many pathways to be differentially regulated between males and females. And one very interesting pathway that we found was the drug metabolism pathway. This pathway was more strongly targeting females, which we can see here by the purple edges. Another thing that is interesting in this method is that we can also find genes, like the CYP genes, that is not the strength of regulation that is different between males and females, but the set of transcription factors. So for example, these transcription factors are more strongly targeting females, and they're all responsive to estrogen. We validated this finding in an independent data set, and then if we think about it, if we're seeing differences in the regulation of the drug metabolism pathway, we can expect that there's going to be differences in the response to treatment between males and females. So we perform a survival analysis for patients that received treatment, and to make the story short, we found that females treated with chemotherapy, the ones that have higher regulation of the drug metabolism, they also have higher overall survival. So with the test for colon cancer, we also have a study with healthy tissues in the GTAX, and I can talk more if anyone is interested. And they're both, let me see. Oh. They're both available on the bioarchive, and the colon cancer study should be published in Cancer Research soon. So, thank you.
Any questions? Yes.
So I'm particularly interested in what I'm calling translational, backward translation, which is it's usually from bench to bedside. But looking at bedside to bench. So for example, we've just published a meta-analysis showing that in various cancer types, looking at immunotherapy, which is a hot topic, that the magnitude of the benefit of immunotherapy is twice as large in males as it is in females. So I wonder if we can talk later about whether some of your research might explain that. And in more recent studies, it turns out if you give chemotherapy to both groups, the females recover. That is, they gain from the immunotherapy. So it's a very complex mechanism, and some of your work might inform why the observations we're seeing in the clinic might have observed.
That is very interesting, and I think we
It's in Lancet Oncology, so I'll talk to you about it.
so we can talk about it more later, and I think so. So just like in colon cancer, it is similar. So the females, there is one paper showed they have better treatment response for chemotherapy, but we don't really know why. And then by using this systems-based approach, we don't only pinpoint a specific genes. What we're seeing is difference in the regulation of a whole pathway, and we can definitely talk more. Yes.
All right, thanks. So next up we have Junwei, one of our new faculty members in the department.
So I'm going to talk about how to read mind, and because although I do not have too much time, so I want to keep my answer as short possible to answer these questions. So my answer is I don't know. So how many minutes do you have? Okay, so since I have some more time, I will talk about the attempts I have made to solve this question in neuroscience. So we have the experiment like that we send some subjects into the MRI scanner, so we can read their minds through when they are inside of MRI scanner. They have a little screen, and they can watch the movie. And the movie is the Sherlock TV series, so we let the subject watch the TV series as a visual stimulation, and we scan their brain to get fMRI data. So on one side you have the data, and each row of data is the activity of each voxel of the brain, and you want to translate the data into the brain network through some graphical model. So the scientific question we like to ask is, on the one side we have the Sherlock movie stories, on the other side we have the brain network from the statistical model. And can we somehow make prediction by the topological structure of brain network and predict what actually the subject's watching, which is somehow like you're reading the subject's mind, and more importantly, can we make this connection replicable in the sense that we want to make it reproducible in a scientific way. Okay, so this is the scientific question we like to answer. And translating into statistical language, we want to combine the word of inference, because we want to make it reproducible, with the word of network of the brain network. So we can see that for the fMRI data, the statistical challenging is that the data is always high dimensional. It has network structure, and it's a cubic structure instead of a matrix structure. And that's why we may involve a lot of computation challenge, like the convex optimization or other massive computation. And then we need to introduce a lot of new theoretical methods to solve this question. So in summary, my research focuses on solving statistical problems with 3 COMS. The first is a combinatorial structure because it has a network structure. And second is a very complicated algorithms and also very complex data structures. So let me get started from the combinatorial inference. So the combinatorial inference we're talking about is to do hypothesis testing to test if a graph is, for example, disconnected versus a graph is connected. We are trying to test if our brain is concentrated or we are all totally distracted. So the network is disconnected. So we can see that this problem is very different from many classical testing we have learned from Statistics 101. The first major difference is most of the classical setting, the parameter is continuous, the networks, they are discrete, so we can no longer use analytical tools. And the second is the testing problem we are testing. Most of the classical setting, we're testing one parameter, which is local. But for combinatorial testing, like testing connectivity, it's always global structure with combinatorial arguments such that this typical algorithm could not be applied. So this is the still challenge. So on the one side, we have very mature, continuous or analytical toolbox about statistical optimization to do the inference. On the other side, we have combinatorial discrete structures to make this world more discrete and hard. And my work is trying to build a bridge between these two sides. And there are many mathematical or statistical methods inside this bridge. And I only want to mention one mathematical method to make a bridge, which comes from the movie Good Will Hunting. So in the movie, Matt Damon is a janitor, and some MIT professor challenged the general math student at MIT about a math question, which they said that it spent them 2 years to solve, and the person who proved it will go to both fame and fortune. And Matt Damon solved it in immediately. Because I'm the new Harvard people, I have to say something like MIT sucks. So I took a screenshot of the problem, and you can see that the problem is actually very easy. Let me to calculate the number of passes inside of this graph, which can be done by a baby. But we have actually a very nice mathematical tool to make a bridge between the continuous world and discrete world, because the problem itself is combinatorial. But the answer is the actual polynomial of adjacent matrix, which is quite analytical. So this is like one example how we make the bridge. Of course, there are more complicated ways in our method, but this is the most interesting part about why MIT sucks and Harvard rules. I've heard Mark Damon, it comes from Harvard, right? Okay. Oh, is that?
Yeah, that's it.
That's
Yeah, but you.
So I have like the final slide about our results. So we make the test about the degree structure, we reject most of the hypotheses, and we find two interesting areas about one is about visual, one about understanding. So we find that actually for the war scene in the movie, the subject is not paying attention, only the visual area is activated. But when Sherlock speaks very fast like I'm doing now, they actually need to pay attention when they need to use the area called precuneus to pay attention. So I won't go to the computational part, but I will holding an informal seminar on Friday talking about these details. So you're welcome to attend, and thank you very much.
Any questions or offended MIT alums? Yeah.
Go ahead, Paige.
It was really Bunker Hill Community College that brought it, right? Not MIT?
And
Goodwill Hunting?
Bunker Hill was where Rob Williams worked, but Goodwill Hunting was MIT. He was the editor.
Oh, okay, all right. So I do have a real question though. So I was a volunteer in a study like this where they measured my functional MRI and I was in there for like 2 hours. I did not get to see Sherlock. I saw a movie of bow ties, polka dot or solid, and I was really claustrophobic and I just fell asleep. I was afraid they would say I had no brain activity. But, and maybe that's true. But what do you do to incorporate auxiliary information about like the age of the subject or the sex or what other things might be influencing their network connectivity in these brain studies? That's
Yeah, so this is definitely a very important factors. Like so far we didn't take into account because there are so many factors and the data is too complicated to take into account these factors. But definitely we'll consider it in the future, yeah.
So when you draw a network, is that correlation in fMRI between the voxels?
Yes, it captures the dependency. It's a partial correlation.
Okay. And then how do you handle conformal mapping of one brain morphology to another?
Oh yeah, there's some like conformal algorithm by unifying all this brain to a standard brain. Standard brain. Yeah.
Is that yours?
That's an exceptional.
All right, any other questions?
Can you go back to your slide with the dates?
Which part?
Oh no, just when you're hosting that seminar.
Oh, yeah. Sorry, I didn't get to skip that part. Yeah, so it's on Friday, starting from next week at 2 p.m. I will send an email to students about that.
All right, thank you.
Thank you.
So last but certainly not least is John, our new chair and professor of comp bioinformatics.
So I'm not speaking today because I'm egotistical. I'm not saying I'm not egotistical, but I'm not speaking today because I'm egotistical. Mullen Wong was going to speak today, and she had to drop out at the last minute. And so last night at 5 o'clock, I was asked if I could fill in. So
That's how it goes.
yeah, that's how it goes. So as the chair, this is part of my job. But what I want to do, I'm going to expand a little bit on what Camila told you about. If you remember, I gave a lightning talk last year, so here are all the important people who actually do the work. I don't do anything. But we have this idea that biological systems are driven by complex networks. The structure of the underlying network is important and informs our understanding of the biology of the system that we're studying. So your network and my network are going to be different. A healthy network and a disease network are going to be different. A brain network and a liver network are going to be different. And the difference in the topology of those networks reflects the underlying processes that are being carried out. So I'll argue that there's no single right network, and people have contested this when I presented it, but I can justify my assertion that this is true, so that each individual network is unique, and that's a big part of what Camila has been looking at, is how networks differ between the sexes. So we have a lot of questions we ask, but we move away from asking, is the network right? Because I don't know how to answer that, but really the question we're trying to ask is the network useful, or does it inform our understanding of the biological systems we're studying? So this is one of my favorite papers from computer science, and it's the basis for a lot of what we do. It's a 1996 paper by Wilbert McCready on no free lunch theorems. And if you read this, it reads in 1997 like an indictment of everything you've ever seen about machine learning. The past few decades has seen an increased interest in general purpose black box optimization algorithms that exploit limited knowledge concerning the optimization problem on which they're run. And they prove a number of no free lunch theorems that demonstrate that you can't just test something on a small sample, but more importantly, and this is what really drives a lot of the work that we do, the results indicate the importance of incorporating problem-specific knowledge into the behavior of the algorithm. And this really resonates with the ideas behind Bayesian statistics, that you want to introduce a guess. In our case, the guesses are biologically motivated guesses. So what's a network? We talk a lot about networks. A network is a way of representing complex associations between entities. And if I have a network like this, Junwei alluded to it, we have an adjacency matrix. The adjacency matrix tells us what's next to what, or if there's a connection in here. When we draw them, we draw lines, and that's a binary matrix. In fact, the matrices that we infer always have some squishiness around them. You can set thresholds to make them positive or negative, or you can actually include the complete structure, the correlation structure, whatever structure you have. But I'm going to come back to this adjacency matrix in a bit. All right? So we have a whole set of algorithms we've built based on the kinds of data we're analyzing. A couple months ago, I did one of these lightning talks before I knew I was ever going to be chair about a method we called Condor and EQTLs. I'm going to talk about the kinds of networks that Camila talked about earlier. So the first is Panda, and Panda tries to solve the question of whether or not we can model gene regulatory processes. There's a paper describing the method. We actually collaborated with Curtis in developing this idea in GC. And it's based on the idea that I'm going to measure RNA, and that RNA is going to come from an RNA-seq experiment or a microarray experiment. To make that RNA, I need RNA polymerase, that's the biology, and this RNA polymerase binding is facilitated by genes that encode proteins called transcription factors. The transcription factors bind and switch the genes on or off. So you can model this as a communications process. The transcription factor is going to send a message to the downstream target. And whether or not that message is actually received depends on two things. Whether the transmitter transmits it and whether the receiver receives it. So if I give you a quiz at the end of this and you fail it, it's either because you didn't listen or I didn't explain it well, okay? But we don't know ahead of time where communications is breaking down. But if I give this whole group a quiz and everybody fails, everybody passes but one person, we know I did my job and you all did your jobs except the person who didn't pass. That's where communication broke down. So if we have a big enough network, we can actually estimate things. So how do we do that? Well, we start with a guess, and our guess is looking at transcription factors and where we know they map in the genome. So we guess an initial network. We take other sources of data and information, and as Camila noted, we use message passing to estimate these two functions, the transmitter function and the receiver function. We update the network, and we iterate the process until the network converges. All right? So what we're going to do is we're going to take two different data sets. We're going to take one subtype of ovarian cancer in this case and another subtype of ovarian cancer. And what we're going to do is we're going to build a network for one and a network for the other. And going back to the hypothesis that different networks represent different phenotypes, we're going to ask the question, where do the networks differ? And the thing we've come to realize from doing this kind of analysis is that the atom of the network is not the set of genes, but the set of connections between them. That what makes one network different from another is how strong these connections are and how the patterns of connectivity differ. There are actually other things we look at, like structure and community in networks, but the easiest thing to think about are these edges, these connections. So we've applied a whole bunch of different, this method to a whole bunch of different systems, and one of the most interesting is looking at cells. But I'm going to take my last 30 seconds as chair. I can do anything I want. And so the question we've been asking is, can we move beyond the single network, right? We take a big group of data, we estimate one network, but really what we'd like to do is estimate a network for everybody in this room. So what I'm going to do is I'm going to take my data set and I'm going to estimate the network for the entire room. Now I'm going to leave Susan out and I'm going to estimate the network for everybody but Susan. And our approach is to use linear interpolation. So these two differ by a little bit. And so what we're going to do is subtract the two adjacency matrices from each other, scale by the number of samples, and just like I did in high school with trigonometry to estimate values between this is really old school stuff, between two different values that are reported in a table. We do linear interpolation and take the difference, add it back to the n-1 network, and get an estimate of your individual network. I put Susan back in, and I take Camila out and estimate her network and do it for everybody in the room. I get a collection of 50 networks, and then we can ask whether or not those networks actually tell us something. And we did this in yeast, and all I'll tell you is in yeast and cell cycle, we actually see the edges oscillate, and the edges oscillate and regulate the cell cycle. So I have a lot more I can tell you. I'm way over time. But people often ask me, are there other questions? Yeah, there are a whole bunch. If you're a student, I can make you work on them and you'll learn things. But then the last thing is, since I'm chair, people ask, well, what's it like to be chair? I've only been chair for a couple weeks, so what does my day look like? Well, if you look, I get coffee, I browse the internet, I spend a lot of time doing that, and then I complain about how much work I have. So, that's it. Browsing the internet is really hard.
Any questions for John? Yeah.
Please.
How do you judge the strength of the interactions in those networks? Is it from variation across individuals or from a single snapshot?
So when we estimate the population level network, what we're doing in a way is we're using message passing to average over the population, right? And that's something we've known how to do for a long time. We take sort of population level averages, we look at correlations, and so we get an estimate of the strength. In our case, we're using message passing to estimate that strength. But the question becomes, how do I estimate your network? And so it took us a long time to stumble on this linear interpolation approach, and it was actually an accident. We had a summer student, Matt Tung, who was working on the problem. We wanted to estimate confidence on the edges. And Matt was supposed to leave out 10% of the samples and run the networks over and over again. When he coded it, instead of 0.1, he put in 0.01 and left out single samples and discovered that even leaving out single samples perturbed the network. And so we went back and looked at that and realized that leaving you out, if we think about this in a really linear approach, every edge in you that might be stronger is going to increase the average. Every edge in you that might be weaker is going to decrease the average. So we figured we could reverse engineer it. And we've actually proven this works exactly for Pearson correlation networks, for mutual information networks, and for, we've demonstrated by simulation that works for lots of other networks. This has been in review for about 2 years. We keep on getting referees who write back and say, well, I don't feel this is right. Kimby, who's been working on this for a long time, her head will explode because we have 6 pages of mathematical proof that it is right, but the referees still don't feel that it's right. So we can talk about that later.
All right,
Not that we're bitter.
Thank you, and thanks to all our speakers, faculty, and students for coming. And a big thanks to the staff who is talking, and the bartender. So, please, everyone, don't go back to work. Hang out for a bit, grab a drink, grab pizza. Yeah.
And if you better not drop out if you promise to give a talk, because I have many more I can do.